Welcome to Chapter 8, Topic 2. So if you thought you got off easy with Topic 1, you did. Chapter 2 is going to be a lot more discussion as well as Topic 3, and these are both going to be pretty new material for you. So make sure you understand them, make sure you take your time through them, make sure you rewind if you need to, and let me know if there's any issues that come up. In this topic, we're going to talk about transcriptional controls and the development of specialized cells. So as always, here's our objectives, and if you have any questions about these, please let me know and we'll make sure we work on them in class. So gene expression checkpoints, as we talked about in the last topic, there are numerous expression checkpoints to help ensure that the genes are being processed properly and that the cell hasn't gotten out of control. And you can see all 6 of them here, and I expect you to understand and be able to discuss this graphic. This is a key graphic in this unit, so make sure you understand that. In this one, we're going to talk mostly about transcriptional controls in this topic. We're going to talk about post-transcriptional controls in the next one. So you're going to see this graphic again and again. And right now, we're just talking about how do we prevent going from DNA to the RNA transcript. That's really the main focus of this topic. So we're going to start with talking about transcriptional switches. What are they? How do they prevent transcription? What mechanisms are utilized to prevent transcription? And then we're going to talk about some examples. This is probably all brand new material for you unless you've been lucky enough to have selected some proteins that we've talked about earlier this unit that are transcriptional switches. So what is a transcriptional switch? Basically, a transcriptional switch is a combination of protein and a specific DNA sequence that is usually a gene specifically. And these proteins will bind to the DNA and inhibit transcription. So that's the most simple way of describing transcriptional switches. Now, an important term that goes along with these are called motifs. What a motif is, it's a structure or a pattern that we'll see in many different protein molecules. It usually will have a whole lot of other things attached, so you can think about it as like a pencil with a big fluffy thing on the end of the eraser. The pencil's always going to be the same, but what's on the end may always be different, and that's the protein differences. So this, the motif is the specific part that's always the same, but remember, it's always part of a greater protein in general. And we've already talked about how DNA and proteins are free to interact when we talked about histones and things like that. So it's important to understand that this is not necessarily a new concept, but that's how these transcription regulators work, is they bind directly into the protein, either the minor groove or the major groove. So the first example of transcriptional switches we're going to talk about is helix turn helix. And in this example, you can see we have 2 alpha helices. And what will happen is this red alpha helices is called the recognition helices, and it will, or helix, and it's going to bind directly to the DNA. What the second one does is the blue alpha helix is going to bind to the backbone of the DNA and provide stability. And so that shows you how this a helix turn helix rather, works. So it's just a kind of an L-shaped almost structure, and it'll bind right there. But remember, this is just a small portion of a bigger protein. This is not the only one, or it's not just these 2 alpha helices in this entire protein. Another example is a homeodomain. A homeodomain motif is a conserved motif of about 60 amino acids or so, and this one actually has 3 helices to it. And you'll see this here where the red is still the recognition helix, and it sits in that major groove of the DNA, while the gray will interact with the minor groove, and the blue once again will provide stability, and you can see that here on this image. This one's found conserved in a lot of eukaryotes. This is a pretty popular example, but once again, remember, just because we see these 3 alpha helices doesn't mean that the that the greater protein as a whole does not exist. It's just this is the homeodomain within the greater protein. So just make sure you understand that. Another example is zinc finger motifs. In this case, zinc is incorporated into the protein. You can see that in this little ball that's between the alpha helix and the beta pleated sheets. Now, generally, zinc finger motifs come in series of 3s with that 1 alpha helix and 2 beta pleated sheets, and it's going to lay in a large portion of the DNA. And so you can see that here where this where the alpha helices will have similar amino acid sequences and bind to similar bases in the DNA, but it's not always the case, and it can actually span quite a range on these zinc finger motifs. So just be familiar with that. And then our last one is our leucine zipper motif. And as I keep saying, I can't stress this enough, these are just portions of a greater protein as a whole. So make sure you understand that this is not the entire protein that you're looking at here. Leucine zippers are interesting because they come in two different types. They come in homeodimers and heterodimers, which means that the two alpha helices, those coils that they make that you can see here in this picture, can be similar or they can be different. And this allows for quite a range in the silencing of the DNA. And so they bind to this DNA and then they inhibit the transcription. And so those are the different ones. Now remember, now that this is our last motif we're going to talk about, the big key here is that all of these are just regions on bigger proteins. Sometimes they could be individual proteins, but for the most part, they're just regions on bigger proteins. And all of these have similar structures, but that doesn't mean that their sequences are the same and they will bind to different genes. Otherwise, we'd have to have massive amounts of motifs. Instead, it's just a structural shape that's similar. So now we've talked about transcriptional switches, and let's talk about some examples in eukaryotes and prokaryotes. And the first of these are repressors. So prokaryotes have something known as an operon, and some eukaryotes do too. It's not all of them, or not many of them do, but some do. An operon is a series of genes that are all transcribed into a single mRNA sequence, and this is useful in prokaryotes because what it allows for is the prokaryote to make one mRNA sequence to generate a whole series of proteins that need to work together. What will happen is a repressor will come into place and it will sit on a specific gene, such as the tryptophan operon you can see here is going to sit on the operator. And what this does is this prevents the RNA polymerase from binding to the gene, and then this can't be transcribed. This is a really good way for proteins to be regulated, is that if tryptophan's present, we don't need any more, so tryptophan just sits there waiting for it to be used up and needed by the cell, and then we'll generate some more tryptophan. So you can see how that works here with this repressor. It's basically a feedback loop, just this time we're looking at the specific actions of it. Another example is an activator. So as you can see here, we have a variety of ways that these genes can be turned on. So in this case, we have the glucose and the lactose, and when both of them are present, the operon's off because we don't need any more. When lactose is present, the repressor is going to sit on there, and it's going to be the lac operon repressor. And the cap isn't bound in this case. In this case, the cap is that activator protein we're looking at. When we're missing glucose and lactose, the cap is going to come on and the repressor is still on, but this still has the operon off because in this case we only want it to activate when glucose is not present but lactose is. When lactose is present, the cap is still there, but the repressor has come off, and the RNA polymerase is free to start the synthesis of the gene. So this is how we go through the lac operon. So tryptophan operon was an example of a repressor. The lac operon is an example of our activators. So just make sure you understand how this works, because the lac operon is only needed when we metabolize, when the sugar for lactose needs to be metabolized, and the cell wouldn't want to make the lactase enzyme unless it's needed. And that's what this example is here for this prokaryotic cell. Remember, these are prokaryotes we're looking at here. So that was simple, right? Here's the harder one, is when we start talking about eukaryotic switches. Eukaryotes, as I said, don't generally have operons. In fact, most of them don't. And so they have these really complicated switches. And we haven't worked out entirely how this works, but we have this enhancer site, and you can see this here on this slide. And it's this eukaryotic activator protein will sit on that, and then the tail of the DNA will flip around and will sit right in that mediator protein that helps the RNA polymerase complex. Remember, we have that transcriptional initiation complex in eukaryotes. It flips around, binds this mediator, and then allows for the transcription to begin. And without this, it wouldn't necessarily start because it's missing that extra piece. So it's important to understand that. Repressors will work in the same way in the eukaryotic switch. It's just much, much farther away. It could be up thousands of base pairs. And we're not entirely sure how this all works and why this has evolved the way it has, but this is it's important to understand that it's not directly where the gene is, like in prokaryotes, where it directly prevents the binding of the polymerase or something like that. Now, we've talked about these in chapter 5 as well. There's another way to prevent the DNA from being read, and that's simply by keeping it too bound up to be read. We talked about heterochromatin, and that's the DNA that is so tightly bound it's never actually read. When we want to have the DNA read, we use chromatin remodeling complexes, which will actually pull apart the histones and allow for the DNA to be read. And we'll also do histone modification with acetylation. And this also can determine whether or not these histones are going to be pulled apart and read, and you can see how that works. The key to this one is that while we have activators, repressors, and the transcriptional switches we just talked about, those are really specific. They target specific genes. In this case, like for example, the chromatin remodeling complex, it's going to open up all the histones. And so it's a bulk DNA read. So it's not going to be able to target one specific gene. So it's important to understand how that works. It's not as fine of a tool to inhibit transcription as it is, as other ones are, or to allow for transcription as it is. So now let's talk about some molecular mechanisms in specialized cells. These are all just kind of examples, because just remember is that all the cells start out the same, but they quickly differentiate. And once they've differentiated, all of the cells that come after the original progenitor cell will always be that way. Because think about what would happen if your liver cells started becoming brain cells, tomorrow. That's going to be a problem. So how does this all work? And this is all through protein control. So let's talk about single protein control. This is an example of glucocorticoid receptor, which is in the liver cells. And as you can see here, it can control 3 different genes. So when glucocorticoid hormone is present, and it's been made by the liver, and it can go and travel to the different genes, and it will activate these genes to be expressed at higher levels. And so you can see how that works. And this is a single protein control. This is simple. And it's not always that one protein will control multiple genes. Sometimes it can control just one. Sometimes it can control more than 3. So there's a whole lot. But remember, this is always part of a feedback loop. We also have combinatorial controls. This is where multiple regulators need to be activated in order for this process to happen. You can see that here. In this case, we have what? 1, 2, 3, 4, 5 different regulators that have to bind to the mediator before transcription will begin. And so this is really important to understand, is that at this point, whatever reason in this example, the gene here would need to have all these triggers before it happens. The cell really wants to control the transcription of this protein. And so it's really important to understand how we can be simple, like for example, the glucocorticoid receptor, or more complex with these combinatorial controls. And remember that these combinatorial controls are like that lac operon I was talking about, but it's just a lot more complicated. And this all gets carried on from the cell to cell. Once one of those regulatory proteins are turned on, it's going to be passed to the next generation of cells and to the next generation of cells. And this is how we go from a single-celled zygote into the multicellular organisms that you and I are. And this is what happens, is as things differentiate, it gets passed on to the next one. So as you can see here, protein 1, when it's expressed at this point in cell division, is going to be carried through for a while. Whereas protein 2 doesn't show up until the other arm of until in both cells, but not until later. And protein 3 is only in a very few of them. And this is how we get to specific cells, such as skeletal cells. And here's an example of the skeletal cells. There's a transcriptional regulator known as MyoD. And what happens is that MyoD, when it's turned on, can change cells into skeletal cells. So if we inject it into other developing cells, it can start leading to muscle development, which is pretty cool. But it's not, this is just one example. This is not how it is for every cell. Not every cell just has this one trigger. But this is just one example that we've seen, that we have found, that shows that. Now, if we were to take this MyoD and inject in that cell, now from here on in, the cells are going to be able to do that. Because remember, every cell has the entire genome. So once we've activated it, it's always going to be there, and it's going to be passed on. And you can see that here on passing on the cell memory. And we've talked about this. This is very similar to genetics too. We've talked about how feedback loops are inherited. DNA methylation is inherited, and chromosome packaging is inherited. And you can think about that because, for instance, calico cats with their heterochromatin, with that bar body for X, their fur color doesn't change in their lifespan. They're always going to have that one patch of orange. It's not going to turn black suddenly one day. And it's always passed on. And so that's an example of how it's passed on as MyoD in skeletal muscle. But as I said, that's just one example, and it's just one we have, but a lot of times it's a lot more complicated than that. And here's another example of how this works, and this is another simple example, and we honestly don't even know how this all works, is that organ differentiation is through these key regulators as well. And in this case, EY is an example of a gene of a protein control that it leads to the development of eyes in fruit flies. So when we inject that into for instance, you can see on the image here, inject that protein into a cell on the leg, it's going to grow an eyeball on the leg. And so this is, as I said, this is a simplified example. Most of these are not this simple. If they were this simple, we would have the answers on that already. So this is just one simple example to show you that not only are these regulators leading to organ development and cell development, but they may change the way these cells and their progenitor cell or their following cells work for the rest of this organism's life. So this is the end of this topic. I know we went through things a bit fast. Let me know if you have any questions and we'll go through this some more in class.